different outcome. We'll talk about it now by one. Oh, okay. Hypotonic. That means well, wow, this is so complicated. Well, the solute concentration outside of the cell is less than the solute concentration inside of the cell. Oh wow, this is a very technical way. Okay, outside of the cell has a lower concentration inside of the cell. Okay, this is easier, just one. Isotonic, they are the same. Outside and inside of the cell, the concentration is the same. Hyper, meaning that outside is higher concentration than inside. Oh, okay, it's the same thing that I told you. Okay, let's take a look at the example of animal cell. When we put the animal cell into the three types of solution we mentioned previously, what will happen? When we put the animal cell in the hypotonic solution. That means low concentrated solution. It can be distilled water. Distilled water is the low concentrated. I would say is if there's no other substances like ion in it, then it is a pure water. Now, of course, water will goes into the cell by osmosis, and then the cell will swell. It's like you are putting a balloon and fill up the balloon with water continuously. When you fill up the balloon with water continuously, what will happen? The balloon will enlarge. Eventually the balloon will burst. This is what it means in here. We call it hemolysis. Well, lysis means breakdown. Remember, hydrolysis is the same. Lysis means breakdown. Hemo means blood, or you can call it red blood cell, RBC. The red blood cell breakdown because it absorbs too much water. It's like you are bursting a water balloon by pumping too much water in it. And then you have isotonic solution. Isotonic solution meaning that you have the water that goes into the cell is the same as the water goes out of the cell. And what solution is isotonic? They call saline. Well, I should call it physiological saline. Physiological saline, what is the concentration? It is 0.9%. We call it physiological saline, 0.9%. If you are admitted to the hospital and then the nurse gives you the IV with the saline, you can see that on the surface of the bag it is printed 0.9% saline. And then you have hypertonic solution. It said that concentrated salt solution, basically you can assume that it's seawater. In this case, the seawater will suck out the water from inside the cell, also by osmosis. So water go out from inside the cell to the seawater by osmosis. That will shrink the cell. Okay, now it talks about different types of fish, freshwater fish and saltwater fish. So for freshwater fish, they live in hypotonic solution. So therefore, water will keep going into the cells of the fish. Then what does the fish need to do? The freshwater fish need to get salt all the time through the gill and also at the same time the freshwater fish, they have to excrete diluted urine all the time. These two mechanism helps the fish to maintain the concentration inside the cell. For saltwater fish, they do the opposite. Because saltwater fish lose water through the gill all the time because of the osmosis, then what happens? They have to drink a lot of water all the time and also actively remove salt so that they have a concentrated urine. So by doing this, it helps them to keep the water inside the cells. So freshwater fish, they have to absorb a lot of salt through the gills, and they have to excrete dilute urine because freshwater fish, water keep going into their body all the time. For the saltwater fish, they have to keep drinking a lot of seawater to replenish the water loss through the skins. They also have to produce a concentrated urine as well to get rid of excess salt. They also get rid of excess salt through the gill. So they try to maintain both type of fish. They try to maintain their tonicity or the concentration inside their cell. Okay, we finished talking about the animal cell on the first column. Now let's take a look at the plant cell. Now let's take a look at the plant cell. When we put plant cell into the same three types of solution, what will happen? When you put the plant cell into hypotonic solution, that means pure water. Water will also goes into the cell by osmosis. But plant cell, they have cell wall. The cell wall will prevent the bursting of the cell. Meaning that, okay, the plant cell originally like this, and then after it absorbs water, it will become bigger. But then because the cell wall restrict how much the cell membrane can expand, then it will remain into that size. An analogy is like, let's say if you are wearing an armor, a metal armor. The metal armor will tightly wrap around your body and your stomach so that you can eat a lot of food, but you have a limit because the armor restrict your belly from coming out. Then you cannot take that much food. When this happens, the cell we call it turgid. Turgid meaning that the cell now is expanded, it's become large, and it becomes very tight and strong. We call it turgid. It is the state when a plant is fully watered. When the plant is fully hydrated, all the plant cells, they are turgid. That's why the plant can stand up straight like this. Isotonic solution. That means it's the same as the physiological saline. The amount of water going in is the same as the amount of water going out. Hypertonic solution. Again, you can put the plant cell into seawater. Then, remember in the red blood cell, the red blood cell will shrink. Similarly, they also shrink in here, but when the plant cells shrink, the cell membrane detach from the cell wall. But some of the cell membrane may still attach to the cell wall. The attachment point we call plasmodesmata. They are the attachment point of the cell membrane to the cell wall. We call it plasmolysed cell or plasmolysis. Lyse, again, break down. Plasmo means the cell membrane. It means the cell membrane got detached from the cell wall. So you can see the textbook figure in hypotonic solution. Okay, the cell swells. The cell mainly absorb to the central vacuole, so that you have something called Turgor pressure. Turgor pressure basically is the central vacuole gets swelled, and then it push the cell to expand, and the entire cell is pressed against the cell wall. This is the Turgor pressure. Isotonic solution, I told you that the amount of water going in is the same as the amount of water going out, so nothing special. Hypertonic, when you put the cell into seawater or when you do not water your plant. When you do not water your plant lose the water in the cell. Then we have plasmolysis. So the cytoplasm shrink. And then the cell becomes soft and wither. Oh, okay, here. So the plant wilt because there's no water, and then the Turgor pressure is lost. That's why the plant becomes soft. So how molecules cross the plasma membrane? I want to highlight this water molecule here. They just don't go through the cell membrane like this. They do not do this like this. I'll put a cross here. As I mentioned before, they have something called aquaporine. They have to go through a special membrane channel called aquaporine so that the water molecule can go through it. Because the water molecule is polar. A polar molecule cannot go through the fatty acid chain, which is non-polar. And then I also mentioned charged ion. They cannot go through the cell membrane. They need a channel, membrane channel, the leak channel, voltage gated channel, ligand gated channel. Okay, non-charged molecule, yes, they can go through this. Large molecule, remember I told you that size matter? If it is too large, you cannot go through the cell membrane. Then what do you need? You need a carrier. I told you that you need a protein carrier, or transport protein, to carry this large molecule to go through the cell membrane. So what can go through the cell membrane? Gas, I told you that. They are permeable. Small uncharged polar molecule. Like urea, water, and ethanol, they are permeable or partially. But of course they require aquaporine, the water requires aquaporine. Ion, they're not permeable. They need, I told you, leak channel, voltage gated channel, or ligand gated channel. Charged polar molecule, well, a lot of this. They are also not permeable. They also require membrane protein. Glucose, remember glucose. Passive transport. Okay, passive transport. Passive transport require a protein channel. I talk about aquaporine, allow water movement by osmosis. Aquaporine is discovered by this person in 1992, Peter whatever. He got Nobel Prize in 2003. It's over what, 11 years. 11 years is considered a short time for people to wait for the Nobel Prize. Nowadays, you have more and more people waiting for Nobel Prize. So 11 years is considered as a short time. You can find aquaporin in all organisms and also in almost all type of cell on the cell membrane. So for the 6 types of aquaporin in humans discovered so far, they allow a relatively faster transfer of water molecule. In some aquaporin, small uncharged molecule can also pass, but no ion. So channel protein. Well, I talk about channel protein. We have 3 types of channel: leak channel, voltage gated channel, and ligand gated channel. And they are for transporting ion or charged molecule. Charged or polar molecule. All these, they have to go through a membrane channel. If your membrane channel malfunction, what can you get? You can get something called cystic fibrosis. Yes, because cystic fibrosis produces faulty chloride ion channel. Normally, this chloride channel will pump excess chloride ion from inside the cell to outside the cell. If your cell has too much chloride ion inside the cell, it's not good. So this channel pump it out. But if it does not pump out, then you have accumulation of chloride ion, and your cells start to secrete mucus, telling you that, okay, I'm sick, I have too much chloride ion. And that's the major symptom of cystic fibrosis. Thick mucus collect in the airway, that means in your lung or trachea, in pancreas or liver duct. So most of the patients with cystic fibrosis, they are European descent, and approximately 4% of the population are carriers. As you can see that inside the lung, the mucus can block the lung for gaseous exchange. Alveoli, they are the small air sac we find in the lung for gaseous exchange. And it can also block the pancreatic duct so that you don't get enough enzyme for digestion or something like that. Okay, last topic, the passive carrier-mediated transport, or we simply call it facilitated transport. Facilitated transport has another name called facilitated diffusion. What's that mean, facilitated diffusion? Basically, it is the simple diffusion that we learn, but facilitated meaning that it requires a membrane protein to transport the substance. So let's take a closer look at the facilitated transport, or aka also known as facilitated diffusion. It is a small molecule and non-lipid soluble. That means they are usually polar. They require a protein, carrier protein. Follow the concentration gradient. So they follow the concentration gradient. What does it mean? Meaning that they move from high concentration to low concentration. Well, just like simple diffusion we talked about, it does not require energy. So we have high concentration on one side and low concentration on the other side. And the molecule will go from high to low, going through this protein channel. So high to low, this direction. They call it downhill movement because from high to low concentration. Example, glucose and amino acid, they are transported in the GI tract into the blood by this type of facilitated transport. The last one, active transport. Active carrier mediated transport, we simply call active transport. Well, we'll talk about it next time. Okay.